Welcome back, fellow mitochondriacs, to another episode of Cancer is a Mitochondrial Metabolic Disease. Hope everybody had a good weekend, holiday weekend, and we are back with it today to discuss further compounds that we have at our disposal potentially for the inhibition or downregulation or the blockade of glycolysis through the enzyme hexokinase. And I had every intention of trying to share this particular compound as kind of a surprise compound the last video. But that being said, I tend to always overestimate um, how much I can get through and underestimate how much information I actually have to share. So I didn't realize I had like an extra like 20 slides to get through. And there was no way that I was going to be able to get through that in like less than 2 hours. So I went ahead and cut it off. So today I want to talk about a compound that I will be very honest, I did not have a great deal of knowledge about, although I've seen multiple diagrams over the last few months, and that is 3BP or 3-bromopyruvate. And it is a kind of an artificial small molecule that has effects on the very enzyme that we have been kind of studying the last several videos as a key protein or enzyme in the relationship between glycolysis and the Warburg effect in cancer. And I just wanted to really do a deep dive on 3BP. I will cut to the chase and say that there is sadly, very limited information about how this is to be used clinically for humans. It is something that has been around for at least 20 to 25 years, probably, in the research community. And you get like glowing reviews from these researchers on the potential of something like this. And then it just doesn't end up in clinical trials, probably for very obvious reasons, most of which it's probably not patentable or something like that. But no matter what, I think that hopefully videos like this and maybe patient interest and kind of a grassroots swell could hopefully turn the tides and maybe get non-industry like governmental organizations to potentially study this, because I think this could be really, really useful for the metabolic approach to cancer. And as as a lot of other things we've looked at so far, including 2DG, but this seems to even have maybe slightly more promise because it is more specific to the cancer tissue than it is the normal tissue, and seems to have seemingly less side effects. So therefore, I think it should be and could be used clinically once we have more information. But I wanted to share not only the background about this compound, to like how it works and why it works, where it works, etc., and give you the potential of where this could go if this was used clinically. But I'm going to give two very somber case reports of this being used in a couple of folks. And although the case reports are somewhat lengthy, I would really like to share it because these are two people's lives who were touched by this chemical, and I just think these people's studies or these case reports are more than 10 years old, and probably are nearly forgotten by the research community, but I wanted to kind of revive their memory, so that's why I'm sharing them. So the first paper I wanted to use to look at this particular molecule was published in February of 2012, and it's titled 3-bromopyruvate 3BP, a fast-acting, promising, powerful, specific, and effective small molecule anti-cancer agent taken from lab side to bedside, introduction to a special issue. And it says here that although the Warburg effect, i.e. elevated glucose metabolism to lactic acid glycolysis, even in the presence of oxygen, has been recognized as the most common biochemical phenotype of cancer for over 80 years. Its biochemical and genetic basis remain unknown for over 50 years. Work focused on elucidating the underlying mechanisms of the Warburg effect commenced in the author's laboratory in 1969. By 1985, among the novel findings made, two related mostly directly to the basis of the Warburg effect. The first, that the mitochondrial content of tumors exhibiting this phenotype is markedly decreased. So what they found is that the amount of mitochondria is markedly decreased compared to the normal tissue. The second finding is that such mitochondria have markedly elevated amounts of the enzyme hexokinase 2 bound to their outer membrane. Hexokinase 2 is the first of a number of enzymes in cancer cells involved in metabolizing the sugar glucose to lactic acid. Later on, it says, finally, the author's laboratory turned to the most important objective. Could they selectively and completely destroy cancerous tumors in animals? This led to the discovery of an experiment conceived, designed, and conducted by Young Co. that the small molecule 3-bromopyruvate, 3BP, the subject of this mini review series, is an incredibly powerful and swift-acting anti-cancer agent. Significantly, in subsequent experiments with rodents, 19 with advanced cancer, co-led a project in which 3BP, or 3-bromopyruvate, was shown in a short treatment period to eradicate 100% of all these cancers. Co-authors' findings, once published, attracted global attention, leading to worldwide to many other studies and publications relating to 3BP and its potent anti-cancer effect. This issue of the Journal of Bioenergetics and Biomembranes captures only a sampling of research conducted to date on 3BP as an anti-cancer agent, and includes also a case report of the first human patient known to the author to be treated with specially formulated 3BP. Suffice to say, in this bottom line, 3BP, a small molecule, results in remarkable therapeutic effects when it comes in treating cancers exhibiting a Warburg effect. This includes most cancer types. So obviously, the author feels pretty strongly that this is a game changer in the terms of treating cancer that exhibit a Warburg effect, which is, as it says at the very bottom sentence, it says this includes most cancer types, as given that the Warburg effect is a hallmark of cancer. So although this paper did include one of the case studies we're going to be reading, I wanted to give further backstory of where this where this has an effect in cancer, so that when we go back to the case reports, we have a better understanding. So the next paper I wanted to share is titled 3-bromopyruvate inhibits pancreatic tumor growth by stalling glycolysis and dismantling mitochondria in a synergistic mouse model. And it says here that 3-bromopyruvate is a promising anti-cancer drug against pancreatic cancer. It exerts potent anti-cancer effects by inhibiting the hexokinase 2 enzyme HK2 of the glycolytic pathway in cancer cells, while not affecting the normal cells. 3-bromopyruvate killed 95% of these pancreatic cancer cells with a 15 micromolar concentration, and severely inhibited ATP production by dismantling the interaction between hexokinase 2 and the mitochondrial voltage-dependent ion channel VDAC1 protein. Electron microscopy data revealed that 3BP severely damaged mitochondrial membrane and cells. We further examined therapeutic effect of 3BP in synergistic mouse pancreatic cancer model by treating animals with 10, 15, and 20 milligrams per kilogram doses. 3BP at 15 and 20 milligrams per kilogram level significantly reduced tumor growth by approximately 75 to 80% in these female mice. It later says that these results provide new evidence that BPV severely inhibits glucose metabolism in cancer cells by blocking hexokinase 2 and disrupting mitochondria by suppressing BCL2, L1, and pancreatic cancer. So a couple of interesting things that I wanted to point out. Number one, the first paper that I showed was from 2012. This paper was much more recent, November of 2022. So the compound is still being studied today, okay, in cancer models, kind of all over the place. Again, still no clinical trials for humans of this, so I'll digress. What I'll say is that, you know, when you hear things like dismantling mitochondria, that is kind of scary for some of us who are mitochondriacs, right? Like, we're like, why would we want to dismantle mitochondria? Well, as we've talked about, when mitochondria are terminally damaged, they will initiate the apoptosis cascade through these caspase systems. So therefore, by doing that, you're effectively reinstating the programmed cell death and inhibiting cell immortality. The other thing I would say is that this is a cell culture. The first, 95% of these pancreatic cancer cell culture, right? And then they took it to animals, mice, and they saw that it had about a 75 to 80% effect. And I think this is very interesting because, you know, I've said this many times, and then, you know, it's very well suggested, and you know, in Seafried's research and others' research, that it very likely needs to be a combination of glucose, glutamine, and oxidative stress inhibition, or not in terms of oxidative stress, you know, enhancement. But my point is that it needs to be kind of a three-legged stool to really have maximal effects, and that still probably is the case. But still, the fact that we're only inhibiting glucose in these pancreatic cancer tumor models for these mice and getting 75 to 80% resolution of the tumor is pretty unbelievable just by hitting the glucose. And remember, these mice are not on ketogenic, you know, feeds. These mice are not getting DON. These mice are not getting HBOT or IV vitamin C at the same time. These mice are getting 3BP, and that's it. So it's pretty unbelievable. Going forward, the next paper was published in March of 2019. It says tumor energy metabolism and potential of 3BP or 3-bromopyruvate as an inhibitor of aerobic glycolysis, implications in tumor treatment. And I picked this paper because it had really wonderful graphics, which we'll see in the next 3 slides. And it says, in recent years, a small molecule alkylating agent, 3-bromopyruvate, 3-BPA, BPRA, you kind of see it annotated differently in the literature. The last couple of papers have shown 3BP as what you'll see. I've even seen 3PO, and in this paper it says 3BRPA as the acronym. But anyway, being an effective glycolysis glycolytic inhibitor has shown great potential as a promising anti-tumor drug. Not only it targets glycolysis process, but it also inhibits mitochondrial OXPHOS in tumor cells. Excellent anti-tumor effects of B3BRPA were observed in cultured cells and tumor-bearing animal models. In this review, we described the energy metabolic pathways of tumor cells, mechanisms of action, and cellular targets of 3-BRPA or 3-bromopyruvate, anti-tumor effects, and underlying mechanism of 3-bromopyruvate alone or in combination with anti-tumor drugs, e.g. cisplatin, doxorubicin, daunorubicin, 5-fluorouracil, in vitro and in vivo. So let's take a look at what they have given us here. So we have our first slide here, which is just a nice refresher for us, looking at cancer from a metabolic disease, right? So let's look at normal, differentiated, non-cancerous tissues that have normal metabolism intact. We see that if you're on a higher, because remember, when we have metabolic flexibility, we can use glucose, we can use fat, we can use protein. But let's just say hypothetically we're on a high carbohydrate diet and we're using glucose for fuel. So that glucose has to go from outside in the bloodstream through these transporters, which we've talked about extensively, and then it has to go through the glycolysis process. And you'll see here that in the presence, and this is what that means here, in the presence of oxygen, the vast majority, we're talking 90+% of this glucose through glycolysis is going to end up in pyruvate. That pyruvate is going to end up going through the pyruvate dehydrogenase complex and getting converted to acetyl-CoA. It's going to participate in the TCA cycle or the Krebs cycle And then those intermediates that are made, NADH and FADH2, are going to contribute to the electron transport chain. And we're going to make ATP through oxidative phosphorylation. Okay, that is normal physiology and normal mitochondrial energy production. You'll see that only in the absence of oxygen or insufficient oxygen or hypoxemia will we have this glycolysis ending in pyruvate, not going to the mitochondria, but going through fermentation to lactate, and then lactate making, in that process, making a much lower amount of ATP. This would be if you're drowning, if you're at a very high altitude, above 10,000 feet. This would be, if you have obstructive sleep apnea at night. This would be if you are sprinting, if you're doing high intensity interval training. But ultimately, what's going to happen is you're going to stop doing those things, or you'll pass away, and you will go back to normal physiology, which is mitochondrial oxidative phosphorylation. Remember, this is normal tissues. For the tumor tissues or cancerous tissues, we see that glycolysis is not behaving in the same way that it would if it was in normal tissue. We see here that it doesn't matter if oxygen is present or absent. We have very defined ways that glucose is being utilized. Okay, number one, 85% of the energy from glucose is going to be made to ferment lactate. Okay, it's going to make energy for these cancer cells. And that lactate is going to be transferred outside the cell, as we've talked about in multiple videos in the past, and create the tumor microenvironment, or the lactic acid tumor microenvironment. 10% is going to be made to make new stuff, right? So the pentose phosphate pathway and biosynthesis of other molecules. Well, there's another slide after this that's going to be kind of highlighting where things go in a tumor model. Okay. Then a very, very small amount is going to get through to make acetyl-CoA. And why is that? Is because there is brakes being put on this energy, this production of energy in the normal fashion, because we have damages in mitochondrial respiration, and we have cellular kind of mechanics that are inhibiting pyruvate to getting to acetyl-CoA by blocking the enzyme that converts that through an upregulation of a protein called pyruvate dehydrogenase kinase. Don't need to memorize that right now. I'm just trying to be thorough when we're looking at this graphic here, okay? But only a very small amount. Even if these mitochondria are intact, which they're not, either, hypoxia factor will either get rid of a lot of the mitochondria, which is why this first author said that they noticed that there was marked decreases in mitochondrial mass, right? The amount of mitochondria are markedly reduced, and the ones that are there are not making much ATP because they're damaged. Okay, this is fairly well known in mitochondrial research and kind of cancer biology, if you're looking for these kind of things. And luckily, folks like Seifried and his group have looked for these things, and they've looked for them extensively. And this is axiomatically true for cancer cells. Okay, now let's go back down to what I was going to tell you about, being a little bit more specific about what's happening here. Okay, so in tumor cells, we have glucose that's being upregulated glucose transporters. We're upregulating all the glycolytic machinery, hexokinase, phosphofructokinase, and all of these enzymes that are necessary to break down glucose, including pyruvate kinase, et cetera. And then what's happening is that through the PPP, or the pentose phosphate pathway, I think in the last lecture, I'm not sure if it was a different part of the world, they call it the pentose cycle. Anyway, we're making new DNA and RNA, we're making new amino acids, and then we're also making, through glutaminolysis, we're making other amino acids, we're making lipids, et cetera, which is why, here's citrate being the kind of the kickoff point to make new lipids for cancer cells, which is why, is giving high amounts of citrate, good or bad, that's up for debate. I would say it's probably more likely bad, but I've seen some conflicting data there. My point is that glucose is being utilized aberrantly. It's not getting into the mitochondria to make energy. It's being used to ferment lactase, or it's being used for biosynthesis of other stuff it needs to make new cells, but it's not being used inside the mitochondria to any degree that would sustain life, okay? This is aberrant metabolism. This is called the Warburg metabolism. I would say the Warburg metabolism plus, because this author did rightfully so add in glutaminolysis, which is not truly part of the Warburg metabolism, but it is seen in most cancers as described by Seyfried and others. So they did bring in glutaminolysis as part of this aberrant metabolism as well, to show really the true and complete picture of what's really going on here. Now, where it gets a little more interesting is we see where 3-bromopyruvate actually does its work. Number one thing I thought was very interesting is that 3-bromopyruvate has a very similar structure to that of lactate. So therefore, it actually enters the cells via these lactate transporters, these MCT transporters. So this, that really makes you think that, okay, is it useful to potentially inhibit these lactate transporters if you're going to be using 3-bromopyruvate? Probably not, right? So therefore, that would be a target that may be off limits if you're going to be using 3-bromopyruvate in the future, because if you were to block these, you would maybe inhibit the effectiveness of 3-bromopyruvate. Now, once it gets inside the cell, inside the cytoplasm, remember, here's the nucleus, right? Here's the nucleus, and here's the mitochondria. We see we're in the free space of the of the cell called the cytoplasm, or the cytosol, which is mostly water. And so 3-bromopyruvate is blocking hexokinase 2, the enzyme that we've been talking about over the last few kind of videos now. But interestingly enough, there's actually quite a bit more to the story. 3-bromopyruvate blocks glutathione to help deal with the excess oxidative stress known to be within cancer cells, and if we were to hit it with oxidative stress, so therefore it actually blocks glutathione. It actually has some effect on G6PD, the enzyme that is responsible for as the rate limiting step to get into the pentose phosphate pathway. That's why I see a down arrow for the PPP. It also has other effects on other enzymes seen in the glycolysis pathway, several of which we will talk about in the past and in the future, excuse me. And therefore, it has a much more kind of like holistic effect on cancer as a whole. It even has effects on glutaminolysis to some degree, which I need to actually kind of look into so that we can see where it works upon. I don't exactly know where it works upon. There's kind of at least two major enzymes that are seen to be blocked by most of the glutamine blocking drugs. That would be glutaminase, which is where DON, mebendazole, betulinic acid, berberine, probably curcumin also have an effect. But there's also other effects on the glutamine dehydrogenase enzymes, et cetera. So I'd be curious to look further into figuring if we know exactly how it blocks glutaminolysis. You can see here it blocks alpha ketoglutarate dehydrogenase. That's this alpha KD, and it also has effects on basically kind of multiple areas within the TCA cycle. And then it also has effects on the actual electron transport chain, which increases oxidative stress and leads to mitochondrial breakdown and likely the activation of the caspase systems that allow for programmed cell death. I mean, this is a very, I don't want to say it. This is a very enlightening slide for me when I actually really go through it with a kind of a fine tooth comb. And it really gives us a lot of hints about the potential of which 3-BPA or 3-BRPA or 3-BP or 3-bromopyruvate has a lot of potential. Again, not to be probably used as a solo agent, although that's how these kind of case studies, they're using it from a metabolic perspective. They're using it in a vacuum, which is sad. But that being said, imagine if you were able to, I don't know, let's say combine a ketogenic diet, fasting, exercise, kind of all things that we know that can kind of shunt glucose away from, a cancer cell, and then add 3BP plus things that we know can have effects on the glutaminolysis system, potentially, maybe, as a secondary agent, maybe fenbendazole or mebendazole that has effects on glucose transporters, hexokinase, multiple enzymes in the glycolysis system, as well as glutaminolysis. That could be a really cool thing. Now, one thing that would be kind of interesting is that you can see that 3BP has an inhibitory effect on succinate dehydrogenase, which, if you remember back I quickly talked about this during when we were talking about fenbendazole, that actually fenbendazole enhanced succinate dehydrogenase activity. And I kind of made a comment that, maybe that could be good, maybe that could be bad. And the reason I say that, I would like to have a better picture here, is that succinate definitely is a pro cancer, kind of what they call tumor metabolite. When it builds up, it can lead to further, it can actually change gene expression and lead to genetic changes, as well as it can stabilize hypoxia factors and lead to kind of like transformation of the cancer cells. So it's not something we want to be built up. So if you were to enhance succinate dehydrogenase and have succinate be cleared out and be converted to malate, that could be potentially good, right? But malate gets converted to oxaloacetate, and oxaloacetate gets converted to citrate, and citrate is what's necessary to create new fatty acids and new lipids for cancer cells. So therefore, is it really good? Is it really bad? I mean, it probably would be kind of a wash. Maybe I would like to study it more, to be honest with you. But my point is that there's actually an opposite effect by 3BP, where it has an inhibitory effect on the same enzyme that would be enhanced by fenbendazole or menbendazole. So that maybe it would be kind of a wash in terms of its effects there. But overall, I think those two medications, those two drugs, could have really synergistic effects. And then if you were to add in some oxidative stressor, whether it be photodynamic therapy, or whether it be IV vitamin C and the right doses, or whether it be hyperbaric oxygen or some other oxidative stressor, I think that could be a really nice synergy. Especially if you were to add it into a ketogenic diet. But again, these are kind of studied in a vacuum, unfortunately. And although that helps us understand the mechanism of that particular molecule in a vacuum, it doesn't really help us kind of like come up with a strategy or of how to be using these things clinically. Now, I pulled this because I thought this was helpful to us understanding how, someone had asked me, I can't remember what compound they asked me. I think it was 2DG, because it feels like it was pretty recent, but it could have been something else. And the question was very good question. And they basically asked, would the tumor microenvironment, the acidic tumor microenvironment, enhance the efficacy of, let's say the question was 2DG, or would it inhibit it like it does our immune system or chemotherapy? Because we know that the tumor microenvironment will, kind of shut down our immunity, and chemotherapy, depending on the type of chemotherapy, because for different reasons. And, I didn't know how to answer the question, and because I didn't know the answer. And so, but when this popped up in the literature, when I was looking through it, and it talked about how 3-bromopyruvate could actually take advantage of the tumor microenvironment being acidic, I thought that was very interesting. So I pulled this. So it said, furthermore, the acidic extracellular microenvironment of tumors favors the stabilization and cellular uptake of 3-bromopyruvate. Indeed, 3-bromopyruvate alone, or in combination with other antitumor drugs, exhibited significant cytotoxicity in multiple tumor cells in vitro, and markedly suppression of tumor growth in animal models in vivo. Notably, no serious systemic toxicity and organ damages were observed in tumor-bearing animals in most situations. Only a few studies reported that 3BP administration produces obvious adverse effects, especially the hepatotoxicity or liver damage, in very few cases, it seems like. And I wonder what doses they were using in those cases. I'll digress. However, in clinical cases, intravenous infusion of unformulated 3-bromopyruvate produced burning venous sensation, and high glutathione levels and serum proteins may also inactivate 3-bromopyruvate. EPR effect may also hinder the retention of 3BP with small molecular size. In addition, 3BP only kills MCT expressing tumor cells, MCT1 expressing tumor cells. The mainstream scientists said that an unformulated 3BP may be harmful in most cases in a clinical setting. This implies an urgent need for novel drug delivering strategies for 3BP in future, among which wafer, liposomal nanoparticles, aerosols, or conjugate formulations may have achieved better tumor specific effects were observed. We considered that these novel chemotherapeutic strategies of 3BP would provide an effective tool in the application of 3BB, as well as other glycolysis-based drugs in future clinical oncology. So what they're saying is that although 3BP seems to really be effective in vitro and in vivo where they're using animals, they're saying that, number one, it has to be a very specific lactate transporter. This is MCT1. And, it's interesting because I would say in most, diagrams that I've looked at, this MCT1 seems to be what's always referred to within cancer models. So I'd be curious to see how many what we call isoforms of these MCTs there are, and potentially what percentage are on a variety of cancers to see, and that could be, again, as a future, instead of looking at cytogenetics or genetic markers in pathology reports in the future, what there could be looking at is, okay, what kind of glucose transporters, what isoform of hexokinase are in these cancers, what isoform of these lactate transporters are in these cancers, what, how dependent of glucose versus glutamine are they in these cancers, like what percent, and then that would help the clinician in the future guide them to know what compounds would be most effective for your particular cancer. Anyway, that's future stuff that's in the probably distant future, unfortunately. But my point is that what they're also saying is that when you give, intravenous 3BP, there are limitations to this medication. Okay, and I would also say that it seems that 3BP, at least in the literature that I've seen so far, seems to be only being used in intravenous infusions, which also limits its use, right? Because you can't just pick this up, on a chem website, right, and start to like take it in by mouth. You're likely not going to have the effects, right? It needs to be IV, and then you're gonna have to say, well, who's injecting 3BP in the modern world? And you're probably not going to find anybody. So therefore, its use is almost impossible. It seems like, although it has great promise. But they're proposing kind of new ways of administration, which I want to get to. Okay, I actually pulled the rest of this paper, which is a really excellent paper, and they kind of give the downsides and the limitations, and they kind of like talk about ways to overcome them. But before we go there, I want to talk about a couple of case reports. The first case report was published in this paper, I believe, in February of 2012. And I only, show this abstract so you know where this case report comes from. And, the paper is titled "A Transitional Study Case Report of the Small Molecule Energy Blocker, quote unquote, 3-bromopyruvate as a potent anti-cancer agent from benchside to bedside." And it says, I'm going to read this case report, so just bear with me. This is a 16-year-old kid's life who had basically a terminal cancer. And I just want to go through kind of this this report, and it looks like it was a hospital in Frankfurt, Germany, where this went down. So herein we present a 16-year-old Caucasian male with a palpable mass over the right upper quadrant of the abdomen. This patient was initially presented to the emergency department of this hospital in this city, which I'm going to butcher, so just forgive me, all Germans who see me in this video. Excuse me, in the Netherlands. It's interesting because this actually says this patient was treated in Germany later on in the abstract, but I'll digress. In the Netherlands, forgive me. His presenting symptom was abdominal pain localized to the spleen. His body temperature was 34.6 Celsius. His first presentation of the blood chemistry revealed high values of AST and ALT, which are liver-related enzymes, as well as GGT, also another liver-related enzyme, indicated liver abnormality and or damage. He was hospitalized immediately and evaluated with a CT and subsequently an MRI, liver biopsy, echo, imaging, and PET scan. The PET scan suggested the patient had very diffuse and metabolically active tumors in his liver. Indeed, all tests indicated primary liver cancer of the fibrolamellar carcinoma form. A second opinion was requested and provided by Erasmus Medical Center in Rotterdam, the Netherlands. It was confirmed that the abdominal pain was caused by a splenic infarct, induced by the pressure of the tumor on the blood vessels supplying the spleen. This resulted in splenic enlargement and some necrosis. The patient had no history of hepatitis nor positive tests for hepatitis. There is no known treatment of this type of hepatocellular carcinoma except for transplantation. And the patient did not fulfill requirements for this process due to the presence of tumor in the choledoductus and spinal lymph nodes. Right after diagnosis, sorafenib had been approved for the primary hepatocellular carcinoma patients as an orphan drug. Treatment with this monoclonal antibody was started, varying from doses from 200 to 400 to 800 milligrams per day. However, lack of experience with such young patients and treatment outcomes was carefully monitored and stopped according to the manufacturer's instruction when the AST and ALT values exceeded the tolerable limits. Initial results with this monoclonal antibody were encouraging. Thus, tumor growth appeared to have halted during the first 2 months of sorafenib application, even with some regression of tumor volume. However, after 6 months, CT scans indicated renewed expansion of the tumor mass and sorafenib treatment was halted. By now, the health condition of the patient had deteriorated. In fact, so far that he could not consume sufficient amount of food to sustain life. On the basis of condition, a duodenal feeding tube was installed and continuous enteral feedings was started. So he got a duodenal enteric feeding tube, and they were doing continuous feeds 24/7. There's kind of two different ways you can do it. You can do bolus feeds or you can do continuous feeds, and they were opting for continuous feeds probably because there was a lot of pressure on the structures, which is why he couldn't eat. The caloric intake was adjusted to 2,000 to 3,000 calories per day. While the patient's health was deteriorating, a contact was established with research scientists, co-authors PLP and YHK at the Johns Hopkins University School of Medicine year of 2008 to learn about the promising experimental anti-cancer agent 3BP. Other contacts were made with TJV, co-author at the University of Frankfurt, for the application of 3BP by transcatheter arterial chemoembolization TACE method delivery. So not only were they just given this like in the veins, they were going to deliver it directly to the tumors versus via catheter, probably via some kind of like specialized interventional radiologist. Permission was obtained from the ethics committee and University of Frankfurt for the use of 3BP in this young patient with this very specific type of hepatocellular carcinoma. The patient's parents signed the necessary consent forms and the patient agreed to the treatment. While waiting for permission from the ethics committee, it was essential to treat patient as soon as possible with currently approved cytostatic by the TACE method. So they basically use conventional chemotherapy via the same method they were going to use via the 3BP. And the first TACE was performed with gemcitabine and cisplatin on January 26, 2009. After delivery of these cytostatics, the blood vessels were temporarily blocked via this embocept. Then the tumor locations were viewed immediately by infusions of lipidol, followed by a CT scan. Due to the cytotoxicity, the first and last of these chemo drugs, due to cytotoxicity, this was the first and last use of these chemo drugs. So basically, because of how toxic these drugs were used, they only use them one time. Likewise, the use of lipidol was not continued during the 3BP treatment due to possible interactions between that medication and 3BP. 1 month later, permissions from the so it took a month for them to approve the ethical use of this medication, despite there being no other treatment available. Okay, that's how messed up this is. 1 month later, they got permission. The patient was treated immediately using the same catheter directed method with the specially formulated that was patented and proprietary BPB twice on the first day of treatment in February 26, 2009, a total dose of 250 milligrams. More clearly, the delivery bolus, meaning the rapid push of the entire dose into the artery in just a few minutes. Therefore, 3BP delivery can be called a transcatheter intra-arterial bolus injection, followed by a brief embolization with this embocept. The patient did not experience any discomfort during the first treatment of 3BP. The only discomfort was caused by the fact that the second infusion took place 1 hour after the first, and the local anesthetic was becoming less effective. So it probably is painful, basically, they're saying, but they didn't really mention this. They gave a local anesthetic before the first treatment, and they didn't give us another. They didn't give another dose of the anesthetic before, so therefore there was some slight discomfort after the second infusion. No other adverse effects were noted during the normal 4-hour recovery period, and the patient left the hospital at 6 p.m. The patient felt very hungry and wished to go to dinner. It appeared that the treatment had increased the patient's appetite. This was an initial sign of hope for relying less on the enteral feeding, or the feeding tube that he had. Indeed, the 3BP had helped the patient consume sufficient calories. Due to the large tumor burden at the presentation, it was determined that to try to obtain very high doses of 3BP in the first treatment month, at the second treatment was scheduled 2 weeks after the first, and the third 4 weeks after the first. So basically, they got a treatment every 2 weeks. After the second application, they used 128 milligrams at that time. That was about half the dose of the first one. They used 250 milligrams, which went without any acute problems. The patient started to get disturbed. 4 days later, the patient went into hepatic coma and was hospitalized with an elevated level of AST and ALT. Pre-existing ascites, or ascites is accumulation of fluid in the belly, basically, or in the peritoneal space, and edema continued to be a major problem for the patient. And despite daily exercise, his mobility continued to be limited. However, during the summer of 2009, he started to regain his physical strength and went out in his wheelchair regularly to the promenade. So because this case report was so long, I went ahead and cut some of it short, because he ended up kind of getting better from the initial kind of issues, and he was able to kind of continue the treatment for a longer period of time. But what it's saying here is that there was a continued ascites and edema. It became a major problem for him, and he started to kind of regain function over time. In addition, the patient was celebrating his 18th birthday, so he actually made it to his 18th birthday, which was told that he would never meet his 18th birthday. And actually, matter of fact, I think it says later on, it said that he would never make it to his 17th birthday, but I'll digress. And this is where this photo was taken on his 18th birthday. The photo was taken in figure 5. At the end of October of 2009, as part of the discomfort deciding was removed, a CT-guided puncture about, I'm guessing it says 1 liter. I mean, they have a lowercase, it looks like another 1. It could be an L. Usually we use an uppercase for liters. I don't know if it was 11 liters, or because that would be extreme amounts of ascites, or if it was 1 liter. And it doesn't seem like 1 liter of ascites for most people would be considered discomforting. So I don't exactly know how much ascites was removed. And sent to pathology to lab for cellular analysis, an outcome of an increased lymphocyte count with some mesothelial cells, but no malignant cells were obtained from the ascites, which is good, because it means that the cancer was not, in the peritoneum. A lot of times when you have ascites in cancer, it's due because there's cancer cells in the peritoneum, and that ascites is what they call malignant ascites. In this case, it was not what we call malignant ascites. The lack of tumor cells in the ascites suggested the tumors in the liver were encapsulated and dead. In December of 2009, the edema and ascites were becoming more problematic. Liver functions were overloaded due to rapid destruction of the tumor cells, resulting in the liver's insufficient detoxification. So basically, what they were hypothesizing is that because 3BP was so powerful at killing cancer cells, it overwhelmed the liver's ability to actually detoxify. What it's saying here is that despite regeneration of healthy liver cells, this process apparently could not compensate adequately for the rapid destruction of tumor cells and could not detoxify the dead cancer cell debris fast enough. Consequently, the blood albumin levels decreasing. The patient passed away 2 years after the first diagnosis due to overload of liver function. So basically, he died, unfortunately, of hepatic failure, it seems like, or liver failure. The patient was able to survive much longer period than expected with improved quality of life, which was clearly attributable to the treatment with 3BP. So although this poor kid who should never have had cancer to begin with at 16 years old, although he passed away, he lived a lot longer than they expected him to. The experts expected him to. And it says here in the figure 5 underneath it, I pulled the graphics. It's text, and it said, "Fighting against immortal cancer cells with immortal spirits of the phoenix." I'm going to butcher his name, so I don't even want to butcher this kid's name. And 3-bromopyruvate. The photograph of the cancer patient in this case report was poised with a novel anti-cancer agent, 3-bromopyruvate, and his favorite legendary immortal bird, the phoenix. The picture was taken on his 18th birthday, September 9th, 2009, at his home in the Netherlands after his 7th treatment with 3-bromopyruvate. This was a very special day, as he was told he would never make it to his 17th birthday. In the morning of this special day, the parents, I cut it off because it was such a long, graphic, but I just thought this was something that has pretty much probably been lost on the research community. And, I'm kind of wanted to, when I saw the picture, I was like, I gotta memorialize this poor kid, because he bravely fought, with all that conventional medicine had to offer, and then was able to, bravely fight with what, was experimental and required, an ethics board to deliberate for a month to allow him to do that. And when he got it, although it was not a smooth, I don't know what the word I want to use is, but it wasn't a complete, happy story. That's obviously at the eye of the beholder. And he was able to live another 2 years with a better quality of life than was expected, for longer than was expected, and make it to his 18th birthday and have a celebration with his family and friends in his home. So that was the first case report. The second case report was published, and this in this paper a couple of years later, and it was titled "The Safety and Outcome Treatment of Metastatic Melanoma Using 3-bromopyruvate: A Concise Literature Review and Case Study." And I have another case report from a 28-year-old male. So a 28-year-old male weighing 60 kilograms presented to the first time with a hard, painless mass in his left forearm on January 2012 and was admitted to Sohag Medical Institute. A sample collected from the mass for biopsy and histopathology indicated melanoma. The mass was surgically excised, and the patient was discharged 2 weeks after admission. Regular follow-up was done monthly. In July, so this is 7 months later, July 2012, there was a local recurrence where a progressively hard nodule was felt in the left forearm at the site of the excised mass. The patient was also presented with pain in the back and left chest wall. A chest X-ray and CT revealed a progressively growing left pleural, basically a lungs, the skin of the lung, or the kind of like the skin of the lung is probably the best way to say it, mass, which caused destruction of a wide portion of the left lung, resulting in its collapse and shifting the heart to the right dextrocardia. The patient was treated with bisphosphonates. Pain in the chest wall and back was intolerable. The patient was treated with non-steroidal anti-inflammatory drugs, ibuprofen and diclofenac, which were insufficient for pain management. As the mass of the left chest wall was continuing to grow, a hard metastatic mass, 5 centimeters by 3 centimeters, pretty large, bulged outside the chest wall. Local radiotherapy, which entailed a specific dose and fractions 5 days a week using a linear accelerator, was applied to the bulging mass. The patient sought medical advice at the Department of Medical Oncology and Nuclear Medicine at the Sohag facility at Sohag University in Egypt. And the patient was admitted September 13th, 2012. Immediately after admission, the initial evaluation revealed that he had dyspnea, or shortness of breath, or orthopnea, where you have shortness of breath when you lay flat, hypotension. Well, that would be borderline hypotension. His blood pressure was 90/60. Anorexia, lack of appetite and lack of oral intake. Generalized anasarca, or edema of your entire body. Rightward shift of the apical beat, which is where his heart was being pushed over by the tumor, and bulging metastatic mass to the left chest wall, and painful regions over his left chest wall and back. Laboratory evaluations confirmed low albumin, low protein, anemia, along with normal kidney and liver function. The patient's whole left lung was destroyed by lung metastases. Per the CT scan, with no air entry, he had no air going into the left lung. He was completely dependent on the right lung, which is why he was so short of breath and had no tolerance to even lay flat in bed, the orthopnea. Thus, the patients remained on an oxygen by mask when necessary. So he needed help with oxygen at some times. Radiographic evaluation revealed a collapsed left lung, pleural effusion on the left side. So the entire lung cavity was filled up with fluid on the left. It's called a pleural effusion. And the metastatic mass, the left chest wall, was causing the mediastinum to go to the right, or the central contents of the chest was being pushed over to the right. The metastatic mass in the left chest was causing, sorry, I said that. Ultrasonography guided aspiration failed to remove the fluid, with little of the hemorrhagic effusion was aspirated. So not only was it cancer likely, it was also bloody as well. The intercostal tube was not used. So because they weren't able to get any fluid out using the what we call a thoracentesis, ultrasound guided, they didn't put a chest tube in because they probably thought it was no use. The patient did not receive melanoma related treatment, but did receive supportive treatment in the form of salt-free human albumin infusion, diuretics. They were just trying to treat his anasarca and his swelling all over his body. Tonics, I don't know what they mean by that. Non-steroidal analgesics, hemostatic agents, Capron, and 1 unit of blood because the guy was very severely anemic. The patient asked for treatment with new lines of chemotherapy for his current status and accordingly informed about. So it seems as if, from looking at this text, that he did his own research and he was like, "Look, I want something else. Give me something else that works because I'm clearly like not doing well here with the standard of care." So he inquired himself. This is what's so amazing about the human will and spirit to survive, right? People with cancer, they, not all, but many will go and do extensive amount of research, right? Which is probably how you ended up on this channel, right? Because you wanted to learn as much as you possibly could, and you end up being more informed than most of the clinicians in this area, right? For metabolic approaches. And this person had found this 3BP probably on his own, probably with very little help from the internet. I mean, it's 2012. It's not like it's the same, data age, I guess you'd say. The patient knew about the 3BP, this mechanism of action and possible side effects. And after receiving another approval, so he has to, this is what's even more frustrating, is these patients, they don't have autonomy over their body, what they actually want and can do, right? They have to get approval from an ethics board determining on what you can do. But I'll digress. That's the way it is. And written consent from the patient according to the Declaration of Helsinki. Treatment using 3BP was planned at safe, low therapeutic doses based on previous reports and publishing studies of 3BP. And they kind of gave all the studies they used to determine that. The therapeutic plan was devised to safely benefit the patient, starting at the lowest possible dose, which would be administered through an intravenous drip. This is what I'm saying. It needs to be likely an IV infusion. This is not something you can take by mouth or use, oral liposomes or something like that. It probably needs to be an IV drip, which limits the utility. Unfortunately, you're going to have somebody who's got to have the medication, know how to use the medication, know how it's used, maybe in combination, and have the access to actually put an IV in. I'll digress. The novel route for 3BP administration fractionates were calculated dose. This may be safer than direct intraarterial bolus injection, which was reported to be effective with minimal toxicity to treat liver implanted in rabbits. So basically they're saying that a slow drip, maybe, kind of an intravenous infusion of 3BP seems to be, maybe it could be safer than just like injecting into the tumor like it's been done in rabbits, but also it was done in that last case report for that 18-year-old kid. The patient's general condition was fair, with except of mild to moderate anasarca or generalized body swelling, which was partially relieved with salt-free albumin. That's never the treatment of diuretics and albumin. It doesn't really fix it. The patient also maintained a good urine output. The treatment, the patient had a normal renal and liver functions with no With treatment, the patient had no normal liver and renal function with no orthopnea or dyspnea. So that must have been improved somehow because he had that at presentation, as we saw. Coulter AU analyzer through the automated clinical pathology laboratory at Sohag University Hospital, Egypt. Before treatment, serum LDH levels were high, which reflects high glycolysis rate and energy metabolism in tumor and metastatic tissue. Follow-up serum LDH levels, cells for lactate dehydrogenase is what LDH stands for, levels indicate response to planned treatment with BP3BP. The first, bear with me, I get this as maybe it's dry for you all, but it's very important to see the how this could potentially be used in clinical medicine, but also kind of the pitfalls and how the patient responds. So the first infusion this patient got was administered on September 18th of 2012. Let's just go back here. I believe this guy presented January. So it took 10 months or 9 months, excuse me, to get the first infusion. Based on the reference dose was 2 to 3.5 milligrams per kilogram of body weight, reported by that co-fellow who actually, I think the one who first used that in rodents. The patient, I'm guessing out of precaution, they used a much lower dose of 1 milligram per kilogram, added to 500 mLs of, it looks like it must have been D5W, 5% glucose. It could be D5NS, I guess. It didn't specifically say, or D5LR. I don't know why you would want to add glucose. Maybe to help prevent hypoglycemia, maybe. It seems like it would be not a great idea to give glucose to a person with cancer via the intravenous route, unless there was really a method to the madness, but they did it. Slow drip over 2 hours. They gave basically 1 mg per kg in D5, either W, NS, or LR, over 2 hours. So basically 250 mLs per hour, basically they got the infusion, which is pretty fast. Treatment was tolerated with no anaphylaxis or unexpected adverse effects. Members of the treatment team attended bedside the patient during the 3BP infusion, and measures for emergency treatment were available. The patient only had the only adverse effects was mild to moderate burning sensation at the infusion site, which was decreased upon slowing the infusion rate. No phlebitis or irritation of the veins, local inflammatory reactions, or allergic reactions were encountered. At the end of the infusion, the patient was in good general condition, lying comfortably in bed and was able to sit, stand, and walk. He went to the toilet and appetite improved. So what did we learn from this first infusion of a lower than likely needed dose of this and given with glucose and probably through a peripheral IV, although it doesn't say it, is that there were really no side effects. There was a little bit burning. We have burning. If you give IV potassium or some other things, you can have burning, and it doesn't mean that it's necessarily bad. I wonder if it was through a peripheral IV or through a central venous catheter or through a port. That would be important to know whether or not where he was getting this immunization through. I think that would be helpful because if, generally we would maybe want central access to help prevent maybe some of that burning sensation. However, despite the burning sensation that it had, that was considered mild to moderate, there was no local inflammatory reaction or inflammation of the veins. So that's kind of a good sign. And he was feeling good after the infusion. The next day, the results were promising. The LDH or lactate dehydrogenase decreased moderately from 4,284, which is quite high, to 3,126. Both renal and liver functions were normal. No metabolic abnormalities were recorded with regard to arterial blood gas, serum glucose, serum uric acid. The blood cellular counts were normal indices without evidence of hemolysis or hemolytic anemia. Bowel habits were normal. And after using 3BP, pain at the chest wall was controlled with a fentanyl patch, basically. All right, moving on. So after this walls of text, I have one more slide, and that's it about it. Okay, I promise. 4 days later, his serum LDH started to rise again and reached 43.53. The patient received a second dose at that time at 1 milligram per kilogram at normal saline only, no sugar this time. Interesting. It was administered via a 3-hour infusion. So they slowed the infusion down from 2 hours to 3 hours, and they gave it in an NS instead of D5, whatever they gave it in. And then treatment was tolerated, and the patient had little burning cessation at the infusion compared to the first time. So slowing the infusion seemed to really help at the infusion rate. As the infusion rate was lower, there was still no any, God forbid, any anaphylaxis, which is like a life-threatening, allergy, or any inflammation of the veins were noticed. Over the next 10 days, the patient received 6 doses of either 1.5 or 2.2. So the first 2 doses, they gave 1.1 milligram per kilogram and 500 cc's or mLs of something, either D5 or NS, and over 2 to 3 hours. And then the next 6 doses, they gave 1.5 to 2.2 milligrams. So they increased the dose in the same amount of NS over this intravenous drip. All laboratory evaluations revealed normal liver and renal function. So there was no hematologic impairments such as neutropenia, low neutrophil count, or hemolytic anemia, or the breakdown of red blood cells. The patient's general condition was stable upon 3BP treatment. Serum LDH was around half of the initial presentation and did not fall into the normal range. Okay, this is where, if I'm not mistaken, this is where it gets a little interesting. Let me double check here. Did they do it in this study? Yes, this is. Okay, good. So this is where it gets very interesting. And I want to give a shout out to Jane McClellan, because I'm nearly certain, and I've only read her book once, so I can't say I'm an expert on it, but I'm nearly certain that she has talked about the use of paracetamol, or basically acetaminophen, as part of the starvation of cancer, right? And I have to go back to her like pathway diagram and even look in the literature myself to see what she and others were using it for. But in this case, what they were using it for was to deplete glutathione. Okay. And because what they were thinking is that, this seems to be a very promising, treatment. The guy is tolerating it just well. His liver-related enzymes and LDH have plummeted in terms of, and there's been no seemingly like either clinical signs and symptoms of any problems, nor any like biochemical, signs and symptoms of problems, looking at all his laboratory studies, et cetera, right? So like, how can we make this more effective? And if you remember back, you probably don't, because I kind of said it in passing. But one of the things that one of the authors was saying was that it was limited by if there's high amounts of glutathione present in the bloodstream, because that will kind of eat it up. And so what they're doing is they're going to give paracetamol as a safe glutathione depleter. Just makes you think about how much you want to be taking glutathione if you're a healthy person, right? Probably not, paracetamol, but I'll digress, which is why I'm not a huge fan of Tylenol in general. But anyway, so what they use is it says, let me read it before I try to paraphrase. On October 5th, 2012, the patient started treatment with paracetamol, a safe glutathione depleter. In the oral form, 500 milligrams tablets, taken twice every 8 hours for 3 consecutive days. It's kind of a lot. That means they're getting about 3 grams of paracetamol during a day. That's a pretty decent amount. Definitely don't want to go any higher than that, because that could cause significant problems. But anyway, I'll digress again. By the next day, the patient received 3BP at 2.2 milligrams per kilogram, added to 500 mLs of NS, or normal saline, administered over 3 hours for 3 consecutive days. So they're depleting the glutathione for 3 days using paracetamol, and they're getting 3 days sequentially of a higher dose of 3BP, which was tolerated without any anaphylaxis or unexpected adverse events. Again, clinical follow-up revealed good response to treatment, as evidenced by decrease in pain with this duragesic dermal pain patch, with moderate improvement of his appetite. Mild lower limb edema persisted, or swelling persisted, with controlled by diuretics. Renal and liver functions were within normal limits. There was a sharp decrease in his serum lactate dehydrogenase from 4,300 or something like that to like 1,800. And then on the next day, it was 58. And the next day, it was 12. It basically normalized the amount of lactate dehydrogenase. So it like halved on day 1. And then I don't even know, like you'd have to do the math, but it like darn near normalized, and did normalize on day 3. 3 days of this treatment with you can just see the power of synergy, right? When you understand like the Achilles heel of cancer, glutathione and glucose and glutamine, and that is the basic premise for how to attack the problem. And this is I'll be honest with you. When I read this case report, I was really encouraged in some ways how simple this was. And it showed such a profound effect by using just 2 of the 3-legged stool, really. The treatment was stopped and the patient was in fair condition apart from an intercurrent chest infection and mild to moderate lower limb edema. Okay, so this is where it turns to the dark side. Okay, so the treatment is working for the cancer. Okay, the problem is the guy is in bad shape. Okay, remember at the time of like presentation in July, the guy had half a lung. His heart was pushed over to the side. It was filled with blood and fluid, and he required oxygen. Right, the guy was not in good shape, unfortunately, when this all started. So although we're seeing like really amazing effects at the treatment of the cancer, unfortunately, the damage to his organism was kind of done, right? So that's why this turns for the worse. And this is going to happen for anybody who's in a hospital for a long period of time. You can get like hospital associated infections, right? Well, this guy had he was in good condition, but he started to develop a chest infection. I'm guessing they mean a pneumonia or something like that, right? The chest infection manifested with fever, cough, respiratory distress, dyspnea. Chest examination revealed there was no air entry in the left side of his chest, or the previously described noted destruction of the left lung. And complete blood count revealed a moderate leukocytosis, or a white blood cell count elevation with neutrophilia, or a neutrophil elevation as well, consistent with a likely bacterial pneumonia. So he's got fever, he's got cough, he's developing a pneumonia. He had the same kind of findings on the X-ray or the CT or whatever they did to show there was previously destruction of that lung. And he's showing now he's probably got signs of sepsis, right? He's got a fever. He's got an elevated white count. Bacterial pneumonia was considered in light of immunocompromise due to malignancy and borderline hypoproteinemia. The patients received intravenous antibiotics, broad spectrum antibiotics. The patient's blood pressure was 90/60, and renal and liver functions were normal. So why that's important is because the way we classify sepsis is based off of either you have the initial signs and symptoms. So he had fever and white blood cell count. So we probably had sepsis, right? And then they were looking at for evidence of end-organ dysfunction. The renal and liver functions were normal. It's important to know that. And his blood pressure was 90/60, which is right at the cutoff to be in severe sepsis. Hypoalbuminemia and hypoproteinemia were persistent, mostly due to anorexia and nutritional deficiency. So because this was also very long, I cut it down. So the guy ended up being admitted to the ICU. He ended up having to be on a dopamine infusion in the ICU where his blood pressure was maintained, we call those pressor medications. So he had what they call septic shock. Urine output increased. Fresh urine was voided in the urine collection bag. Liver and renal functions were normal. Echo revealed the mass. So this poor guy, man. So the mass in his chest was actually pushing into the left ventricle of the heart. Okay, that's probably with a moderate pericardial effusion, which means it's got fluid around the heart, kind of pushing on the heart. The diagnosis was impending cardiac tamponade. That's a life-threatening condition, which may have been secondary to metastases from that leftward, due to that leftward shift, as well as severe nutritional hypoproteinemia. Overall cardiac movement was normal. Patient maintained on treatment with antibiotics, dopamine, dobutamine. So he had cardiogenic shock as likely as well. And diuretics in the intensive care unit. Edema in the lower extremities decreased, and the blood pressure was stable at 100/60. Leukocytes increased despite broad spectrum antibiotics. Liver function tests, including AST, were within normal limits. Slight elevation. Basically, the guy died of what did it say? The patient was in respiratory distress, hypoxemia, despite giving him oxygen, probably subsequently intubated, and he passed away of low oxygen levels on October 12th. And if I'm not mistaken, the major success was happening on October 8th, which means he rapidly deteriorated. Right? I mean, he went from October 8th, where he was fine, basically, he had a mild, chest cold, or probably an early pneumonia to, basically, I'm sorry, that was the 9th. So essentially 3 days later, he was dead from hypoxic respiratory failure in the ICU on pressors. So what can we take away from this unfortunate? I mean, the guy was 28 years old. Maybe he was 29 at the time of death. that doesn't always go, like we want in clinical medicine. When you think you're winning one front of the war and the battle, something else takes over. Just being in a hospital, unfortunately, is a risk factor for death because you're exposed to a lot of, bugs. Even the best we do to try to keep things clean and sanitized and sterile, we actually grow out kind of the nastiest bugs in a hospital. Carbapenem resistant bacterias, methicillin resistant bacterias, et cetera. And that's just that's what happens. And when your body's already weakened by a tumor eating half your lung, and with these giant pleural effusions he had, and not able to breathe, and probably not able to take big deep breaths in, so that can lead to other problems. And the guy was in bad shape, unfortunately, when they started the treatment. They waited 9 months too long. Let's just be honest. They waited 9 months too long. If maybe you can make the argument at least 1 month too long, right? They took him a month to probably get him through that, to get him approved to take a medication that was not approved or that was experimental for a guy who had no real hope with the standard of care. And he paid the ultimate price for that. And I wanted to kind of memorialize these two individuals who unfortunately did pass away from their disease process in some way, shape, or form. But no matter what, they give us invaluable lessons from their stories. And we can see kind of glimmers of hope from these two stories as well. And in the conclusion of this paper, it says that 3BP can be regarded as an antimetabolite, being structured as an analog of pyruvic acid or pyruvate and lactate that can be administered by slow intravenous infusion with minimal hepatic, renal, and hematologic toxicities. Anti-cancer efficacy of 3BP can be antagonized by high tumor GSH content, but can be potentiated on concurrent administration of GSH or glutathione depleters such as paracetamol. Future clinical trials using 3BP and anti-melanoma agent as an anti-melanoma agent and general anti-cancer agent are strongly recommended. Has not happened. Despite their strong recommendation back in 2014, that has fell on deaf ears. There's been no clinical trials on this medication. When the anti-cancer effective 3BP needs to be potentiated, combination with paracetamol may be considered. I just think that it's just so sad that these kind of agents have such potential, seem to be relatively cheap and safe, and have minimal side effects if they're probably done under the proper supervision and in the right dose that we understand so far, and probably in the right time too. So anyways, I'm going to digress for a second here and take a look at the comments we've had. I didn't realize I've gone an hour and 12 minutes going through these case reports, and I hope it was beneficial for your understanding of how these go, how these cases go, even when you're maybe finally giving the right medicine. Thank you for spreading knowledge and hope. You're very welcome. It's not really me. It's these case reports, that's really spreading the knowledge and hope. And I'm just kind of being a mouthpiece at this period of time. Good afternoon to everyone. Good afternoon to you too. Any idea why the FDA shut down one of these doctors' source of 3BP? I don't understand why. I can tell you that, the slides that I'm not going to get to, unfortunately, because we're already an hour and 13 minutes in, is that they are, let me just quickly go through what they found was that there's a bunch of, these are the 13 findings that they studied that are contributing to the lack of tumor efficacy and specificity. And they propose their, ways to improve it, whether it be with liposomal modifications or adding certain things to the liposome to make it more, it's freaking awesome. Actually, if you actually read these, I'll tell you what paper this is from. It's from Enhancing Anti-Cancer Effects, Decreasing Risks, and Solving Practical Problems Facing 3-Bromopyruvate in the Clinical Oncology, 10 Years of Experience. This paper is really, really good. They go through the hallmarks of cancer. They go through the hallmarks of how what disturbs the 3BP effects, and they give their kind of gestalt, but they have we're talking about large paragraphs of text about how they can overcome these things using certain technologies. So you know, part of me, I would say the cynical part of me, wants to answer your question by saying that they just want it to be patented with something that's novel that they can make a bunch of money on. Um, that's probably where I like 80% believe. 20% of me believes that it just does need improvement. And there's new technologies that are being developed, God willing, that are hopefully going to be in the market that can help augment metabolic therapy. I would never want to use 3BP as a standalone agent or any other agent as a standalone agent, but I think it could be used in combination with, God, it's something as simple as paracetamol. But let's say a ketogenic diet and Dawn or mebendazole or something like that with something like an oxidative stress adder. I think that could be a really cool winning combination. It's just that we got to see it. We got to see these potential combinations, and I can come up with 10 combinations in my mind right now, that could probably be effective, but it's got to have to be studied, at least in case reports or at least in, kind of animal models and trying to get that to the bedside, so we can do it and not get in trouble, basically, and that we don't want, we want to help people. And it just seems like it's taking way too long, 10 years. How many people have died in 10 years, from cancer, waiting for this people's paper to come out? It's just really sad. I thought IV vitamin C was an antioxidant. How does it work as an antioxidant, or has it? I'm butchering your question here. How does it work as an oxidizing agent for cancer? Well, Carrie, that is the paradox, the great paradox of vitamin C. So I'm going to have a whole video on it, of course, but vitamin C at low doses IV will act as an antioxidant and likely promote or protect cancer. Any doses of by mouth or liposomal vitamin C is probably a no-no as well, because it doesn't reach the serum concentrations needed to have that paradoxical oxidative effect. But when you have it in the right effect via IV, it actually acts as an oxidizing agent, and it increases oxidative stress for cancer cells. It will break down to hydrogen peroxide in particular. And what these, this is people that are not necessarily interested, but not at all like studying the metabolic effects of cancer. What they believe is that the enzyme catalase which breaks down hydrogen peroxide, is generally deficient within cancer cells. And so they believe that it's that high dose of hydrogen peroxide that leads to ferroptosis and lipid peroxidation and cancer cell death, So that's how it works. I'll have videos on it in the future once we kind of hit all these glucose and glycolysis and kind of the three-legged stool, I guess I would call it, the two legs of glucose and glutamine, which, we've pretty much hit glutamine to the maximum amount, although there are a couple of things that I'd like to probably share about glutamine in the future, but then I'll hit oxidative stressors and we'll go there. Thank you, Paul, for your contribution, as well as Cam's. Any issues with taking EGCG, berberine, vitamin D together? I mean, it's going to be a case-by-case basis, but I can't imagine why there would be as long as you're taking doses within kind of proposed doses, I can't imagine why there would be a problem there. I mean, I can't imagine. As I will say, one thing I do generally recommend is that I'd recommend people get their vitamin D from the sun and not from a pill, and treating to a serum 25-hydroxy vitamin D level. P450 over in the liver overwhelmed, basically. Yeah. The question is, could they have added something like, milk thistle or something like that? It's hard to know, because we obviously, some things we do for liver related damage like that acutely would be like glutathione or NAC, but that would be kind of contradictory to the glutathione, what do you want to call it? Glutathione depleting treatment they were using with paracetamol. So it's really hard to know. It's probably, how do I say this? Probably, God, I don't have a slide pulled up right now, but if you look at how Seafried very eloquently described the method, the madness, it's not about like killing it all in one day, right? It's about like slow pulses of these things. And although I do believe that some of these things don't need to be pulsed, personally, the other method to that madness may be trying not to overwhelm the system with dead cancer cells, and have like a tumor lysis syndrome, or a hepatic overwhelming and liver failure like this unfortunate poor kid had. So maybe that's part of the method to the pulse. Not that you need it, because your system is going to be negatively affected by a glucose blocker being continuous in your system, or a glutamine blocker being in your system, but maybe it's too effective. and it would lead to clinical outcomes that would be less than ideal. Maybe that's the reason for being a little more methodical and slow about the process. Vitamin at low dose, antioxidant high dose, greater than not milligram IDV. Remember, it's grams, not milligrams. It's a gram per kilogram dose, actually, Amy. Have you looked into chlorine dioxide solution instead of vitamin C? I have not. I have not looked at chlorine dioxide. 3BP sounds so promising. Thank you. Yes, it does. It does sound so promising. It sounds very promising. We need some of this real science to get out and make it to the bedside, right? Thanks, Doc. Good talk. CDO, oxidative stress contributor. CDO. I'm not sure what CDO stands for. What that acronym stands for. Can you tell me in the chat what CDO stands for? I'm guessing it's a chlorine dioxide. I don't know about that compound, unfortunately. I haven't looked into it. Looks like that's all the comments. So with that, I'm going to leave us off on this slide as being the last slide again, so that we can all see all of the hexokinase 2 inhibitors that we've been able to go through over the last several videos. You can see that, things that a lot of us are aware of, EGCG, sulforaphane, artemisinin, capsaicin to some degree, luteolin. Here's our 3-bromopyruvate, right? Metformin, fenofibrate, ketoconazole, posaconazole, which I've never used clinically. I've never even heard of it until I had to look it up. But it's just another azole antifungal medication. Resveratrol, curcumin, quercetin, vitamin D, melatonin, apigenin, astragalin, reishi mushroom, ganoderma, oleorupine, membendazole, fenbendazole, and 2-DG. Those are all of the hexokinase 2 inhibitors that I was able to find going through the literature. And as per usual, I'm always very surprised on when we really do a deep dive, what we're able to find. And just realize that we have so many tools in our tool bag. And remember, like a lot of the cool thing about especially the natural ones, although we were able to find out that, 3-bromopyruvate and 2-deoxyglucose have other effects, membendazole, et cetera, have ultra effects, metformin have multiple effects as well. A lot of these natural compounds, some of my favorite being, EGCG, quercetin, vitamin D, melatonin, of course, they all have so many different effects that are beneficial to this process, which just goes to show that like having a polyphenol rich diet is such a great, kind of way to prevent disease, but also having the major internal guardians, what I consider the major internal guardians of vitamin D and all of its beautiful analogs, and melatonin and all of its derivatives, kind of going through your body continuously by having adequate sun exposure, infrared exposure, as well as darkness at the right period of time at night, are really showing us the story of why someone would get cancer to begin with, right? When you have, crashed vitamin D levels and your melatonin levels are crashed because you're up on your phone all night, or watching TV and not getting outside because, the dermatologist told me not to get outside because my risk of whatever is going to be higher, which is likely not true, then, you can see how the human organism is is unprotected from its most serious conditions, one of which being cancer. So anyway, if you like videos like this, please like, share, subscribe. Thanks for bearing with me with the case reports. Hopefully you can participate by listening to these case reports and maybe even looking them up and kind of immortalizing these two young individuals, a 16-year-old to an 18-year-old male and a 28-year-old who died in horrific ways, likely of a kind of a liver cancer and a melanoma. And until next time.